Thank you for introducing me. Yes, my name is Juyong Park. I'm now doing postdoc at Stanford Medicine. Today I'm going to talk about my representative PhD work about roles for translation regulation in gastric cancer pathogenesis. Let me introduce myself briefly. I did my undergraduate and master's in Seoul National University, where I did plant biochemistry research. And then I worked at KIST for my military service. And then I did my PhD in biology at Stanford for molecular biochemistry and cancer biology research. And now I'm doing a postdoc in Stanford Medicine to generate organoid model for immuno-oncology study. This is my outline for today. My talk starts from introducing lysine methylation and methyltransferase terms MYT5 and some gastric cancer intro, and then talk about how I identify the SMIT5 catalytic activity and its biological and etiological roles in gastric cancer pathogenesis. And finally, talk about SMIT5 as a novel therapeutic target for gastric cancer. I'm introducing here the well-known theory of biology, the central dogma. I hope many of you here probably heard of this. In 1956, it was originally proposed by Francis Crick, the person who discovered the structure of DNA. He states that the genetic information flows one direction from DNA to RNA and then to protein molecule. So actually, does it mean that one gene can make one corresponding mRNA and one corresponding protein? So if mammalian genome has about 20,000 genes in the genome. So this number of genes can make the corresponding number of the mRNA and the corresponding the number of proteins. But the later research showed that real biology is not that simple. In fact, RNA and proteins can undergo biochemical modifications to generate many different forms. So the 20,000 genes are being transcribed, and then they undergo post-transcriptional RNA modifications, and to create over 100,000 variations. And then post-translational protein modifications can make further variations for over 1 million different forms. In particular, these post-translational protein modifications ensure PTM greatly increase the functional diversity of proteome and play an important role in the number of cellular processes. So among the PTMs, my former PhD lab, the Gojani lab, has been particularly interested in this protein methylation. Let me give you a brief intro of lysine methylation. Proteins can be methylated at the lysine amino acid. At the lysine has the epsilon nitrogen with protons. And a lysine methyltransferase, in short KMT, can add one methyl or two or three trimethyl moieties replacing the protons on the epsilon nitrogen. So creating these four distinct unmethyl, monomethyl, dimethyl, and trimethyl, four distinct states on the lysine residue. And this is a reversible reaction, can be reversed by lysine demethylase, in short KDM. So why do we study methylation? Methylation was first discovered in the flagella protein of Salmonella. And I'm introducing this to the Nature paper 1959. A fun fact is that this is the entire Nature paper in 1959, just one figure. So but my Nature paper this year has 100 of figures. It means there is a vast inflation in science since then. Anyway, in 1964, methylation in histone proteins was first discovered by Kenneth Murray at Stanford. And with the discovery of the first histone methyltransferase in 2000, histone methylation became a key topic in chromatin biology and epigenetics, leading to many publications. And with the mass spec-based proteomics, it has been discovered that over 1,000 methyl proteins or events, and it's estimated that 75 to 100 candidate lysine methyltransferase in the human genome. And among these, EGH2 histone lysine methyltransferase received FDA approval in 2020 as the first KMT inhibitor now used to treat sarcoma and follicular lymphoma. So mammalian cells have over 500 protein kinase and regulating a number of cellular processes and often linked to human disease, and now making them important therapeutic targets. And as I mentioned earlier, the first histone lysine methyltransferase was discovered in 2000. And then about 20 years later, there was the first drug going into patients targeting one of the lysine methyltransferase, the EGH2, and now a few more clinical trials. And those KMT enzymes seem to have great connection to disease, but we don't know much about what they are doing. And even if you know something about their activity, we often don't understand exactly what's the KMT activity. So I think there is a great potential to learn about KMT biology and disease, and then potentially interventions based on the findings we can help people, the patients. So among the putative KMT enzymes, one of my PhD project focused on the SMID5, one of the potential lysine methyltransferase. Before I'm going on the SMID5 biology, I will give you a brief intro about what is SMID protein. There are 5 SMID family proteins, which all have mean split set domain. Over the years, these SMID proteins have been published to be histone lysine methyltransferase, but many of them were not reproduced in multiple labs, including our lab. Instead, their physiologic substrate were demonstrated as non-histone proteins. So among these SMID family proteins, SMID5 was classified as histone lysine methyltransferase. Now I'm showing 2 representative the reports of histone lysine methyltransferase of SMID5. But there remain questions like other SMID proteins. SMID5 physiologic substrate could be a non-histone protein, but no one had tackled this before my project. If so, why important to study SMID5? Here I was doing some cancer analysis of SMID5 genetic alterations in gastric cancer patients. And found more than 80% of gastric cancer patients have SMID5 overexpression. And when I look at the patient outcomes, I saw that SMID5 high expression significantly associated with the poor patient survival rate in gastric cancer. And then, interestingly, SMID5 total knockout mice, it appeared they don't care. They are viable based on the MPC data. So it led me to hypothesize SMID5 inhibition maybe has great potential for gastric cancer treatment. So it is important to study gastric cancer because gastric cancer is the fourth leading cause of cancer-related deaths worldwide. Nearly 800,000 deaths from 2020 data. And gastric adenocarcinoma accounts for over 90% of gastric cancer. So I may use the GAC term throughout my talk representing gastric cancer. And once the stage is advanced, it becomes quite lethal. And interestingly, incident rate is much higher in Asian countries. So Asian countries take up about 50% of new cancer case worldwide. but 75% of new gastric cancer case. So there is a critical math need to identify novel therapeutic target to treat gastric cancer, and SMIT5 could be the strong therapeutic target. But before I could answer this number one question, first I need to know SMIT5 catalytic activity. And so just to see if this was the reported activity I mentioned earlier. If this was activity I could reproduce, I just simply made SMIT5 knockdown cells to see what happened to the histomethyl states. Surprisingly, I was not able to see any histomethylation change as they republished. Second, SMIT5 primary cellular location is even in cytosol, not the nucleus. Finally, I was not able to see reproduce the SMIT5 histone methylation activity on either octamer or nucleosome in vitro. So I concluded that SMIT5 is probably not histone methyltransferase, which led me to identify an unknown substrate. So how do I do this? Here I introduce the system that I established to identify the physiologic substrate in an unbiased way. This simple in vitro assay using human cell lysates. First, I took control cell lysates and added SMIT5 protein, and the radio labeled SAM, the methyl donor, and set up the in vitro reaction. And you see, you don't see any noticeable band either upon adding wild type SMIT5 or catalytic dead SMIT5. But when I use SMIT5 knockdown cell lysates, you can see a clear noticeable band only upon adding SMIT5 wild type protein. So this is the only detecting reactions with knockout extract and wild type SMIT5 protein. So I was thinking, because the substrate in wild type probably close to 100% methylated in normal cells, and this band reproduced in multiple different cell lines, and the 10 kD band size was distinct to the either histone H3 or histone H4 protein. So I thought it's likely a physiologic substrate. And then I did mass spec analysis of this protein band, but unfortunately was not able to see any methyl protein. Because this guy might be the low abundant protein or some specific peptide specific sequence might not be detectable in the mass spec setting. So to address these issues, I used a 5-step fractionation protocol to enrich the metal band. And I got down to a very simplified band, the protein sample, and analyzed by mass spec. And the top hit was UBA52. So there are 4 types of ubiquitin encoding genes in mammalian cells. One of them, UBA52, is a fusion protein, monoubiquitin and alpha-40 protein. And after it's synthesized, it's cleaved by deubiquitinase. And the alpha-40, the ubiquitin, monoubiquitin can go into ubiquitin pool, while the alpha-40 makes up the large ribosomal subunit. And interestingly, I found a 30-year-old paper showing that alpha-40 methylated in the lysine 22. So if you look at closely the alpha-40 protein sequence, I saw it cannot be detected in the standard mass spec settings. So I hypothesized that alpha-40 could be the SMIT5 substrate from my previous sample. To test this idea, I did in vitro methylation assay with alpha-40 and SMID5. In the SMID5 methylate alpha-40, but not the SMID5 catalytic mutant. And this shows SMID5 does not have the histone methylation activity, but very strong activity for alpha-40. And if you make the methyl site mutant, K22 mutant, it eliminates the methylation. And then next, to follow up the methylation in cells, I made alpha-40 methyl state specific custom antibody. And I used that antibody here to check the methylation in cells. I saw SMID5 knockdown cells. It makes alpha-40 the methylation deplete. And SMID5 knockdown did not impact neither monoubiquitin or other ribosomal components. And then to the SMID5 depleted cells, I added back either SMID5 wild type or catalytic mutant. And SMID5 wild type recovered alpha-40 methylation, but not catalytic dead mutant, indicating SMID5 catalytic activity is required for physiologic alpha-40 methylation. So the long journey to identify SMID5 substrate was very challenging for my first 4 years of PhD, but now you see my happy face. So we found that we discovered RPL40 is being methylated by SMID5. So I wanted to understand what the methylation is doing. To get insight to this, I did some structural studies using the published human ribosome structure. And the 6th large subunit is now here highlighted in the light blue. And the 40th small subunit is marked in the salmon color. And this study shows that RPL40 position in the A site in the 6th large subunit and adjacent to mRNA channel. So it led me to explore whether the RPL40 methylation may regulate mRNA translation. To test the idea, I did polygenic profiling of gastric cancer cell extract. Briefly, cytoplasm extract was spun in Scrooge gradient. And then ribosomes are separated by their densities, and the mRNA is bound to each fraction of ribosome can be detected by UV observance. Here I'm showing the sample profile. You can see each peak means the mRNA bound to each fraction of ribosome, like 40th, 60th, 80th monosome. And you can see the disome, the 2 ribosome clusters, and trisome, 3 ribosome cluster, and more polysomes. I want to especially emphasize that polysomes are actively translating ribosomes. So most of mRNA translation occurred at the polysome fraction. Here's the data comparing SMID5 control and knockdown. And each peak shows the mRNA bound to each ribosome fraction. When I compare the area under the graph for the monosome and polysome versus the SMID5 knockdown cells, and plot the ratio of the polysome to monosome, the area under the curve. And you can see upon the SMID5 knockdown, the polysome to monosome ratio increased. So this is finding we can make 2 possible explanations. First, more mRNAs are bound to polysomes, meaning more protein synthesis, or less synthesis because polysomes are clumped together due to defective translation. So to test both possibilities, I directly measured protein synthesis in cells. I used here a puromethylation assay, a common method to measure the protein synthesis. In this assay, we had puromycin, a tRNA analog that can be incorporated into the growing polypeptides. And I treat the cells with puromycin for 15 minutes. And in the time frame of the treatment, any elongating polypeptide can incorporate the puromycin, and we can detect the puromethylated polypeptides by Western's. And the intensity of the puromycin is a proxy for the protein synthesis. Here's the data. There's less signal, means less global protein synthesis upon Smith5 knockdown, indicating that Smith5 loss caused a defect in global protein synthesis. And this phenotype is reproduced in two different gastric cancer cell lines. So I postulated this global downregulation of protein synthesis upon Smith5 knockdown due to alpha-40 methylation, because alpha-40 is the physiologic substrate of Smith5. But it's hypothetically possible that this phenotype upon Smith5 loss could be due to alpha-40 methylation or maybe something different function of Smith5. So to test this idea, I made alpha-40 knockdown cells, and there is a huge decrease in neosynthesis of protein. And this phenotype was fully rescued by complementation of alpha-40 wild-type protein, but only partially restored by complementation with a non-methylatable alpha-40 K22R mutant. So indicating that the reduction in translation we saw earlier from Smith5 loss was directly due to alpha-40 methylation. So suggesting alpha-40 Smith5, the methylation pathway regulates protein synthesis. And next, I mentioned earlier heavy polysomes where mRNAs are actively being translated. So I look at the mRNA bound to polysome in the control versus Smith5 knockdown. And I analyzed them by RNA sequencing with a collaborator, Ivan Lab, the McGill. And we found that there were key differences for those guys, 1,500 mRNA species. And functional analysis of those mRNA species identified a number of programs like MYC, ribosomes, and other gastric cancer associated signatures, indicating they are alternatively associated with the ribosomes, means alternatively translated upon Smith5 depletion. And in fact, when you directly look at the protein levels, some of those targets from the signatures, and we saw that protein levels were reduced upon Smith5 knockdown. And this led us to hypothesize that Smith5 regulates gene expression at the level of translation in such a way that might impact gastric cancer pathogenesis. So we found Smith5 methylates alpha-40, and it regulates translation, and it impacts gastric cancer pathogenesis. So dysregulated protein synthesis is a common trait of any type of rapidly growing cancer cells, raising possibility that SMID5 via alpha-4K22 trimethylation may influence cancer cell phenotypes. To get insight to address that question, I first compared the expression level of SMID5 and alpha-4 methylation in non-transformed cells and compared to the multiple different gastric cancer cell lines. And you can see SMID5 and alpha-4 methylation and total alpha-4 levels in cells is increased in 4 different gastric cancer cells than the non-transformed IMR90 or WI38 cells. And this increase is much selectively different than other ribosomal protein alpha-6. So it means SMID5 and alpha-4 methylation may selectively function in the gastric cancer. Moreover, alpha-4 methylation signals increased in gastric cancer patient tissue than human normal stomach tissue. And the signal increased with progressing gastric cancer stage. So I thought that SMID5 potentially has a role for gastric cancer neoplastic growth. To test the role for SMID5 and its catalytic activity in gastric cancer growth, I used a physiologic in vivo model called xenograft to test the tumor growth. And first, I'm just going to tell you about we collaborate with the Pablo Major Lab for the mouse studies. So I prepared gastric cancer cells with plus minus SMID5 and complemented with wild type SMID5 or catalytic mutant. And we did a xenograft assay of tumor growth. And we found that SMID5 depletion inhibited the gastric cancer cell line tumor xenograft tumor growth. But this growth was restored by complementation with wild type SMID5, this red line, but not with the inactive SMID5 mutant, the purple line. And more interestingly, these phenotypes were observed in a gastric cancer patient-derived xenograft, indicating SMID5 affects gastric cancer cells upon its catalytic activity. And then we still wanted to know whether the SMID5 depletion and effect and dependence of catalytic activity was directly due to alpha-4 methylation. So to test that idea, I generate gastric cancer cell line and generate the alpha-4 knockdown and complemented with alpha-4 wild type or alpha-4K22R mutant, which cannot be methylated by SMID5. And we saw that the growth of wild type alpha-4 complemented tumor growth fully rescued compared to the control levels, whereas the xenograft tumor growth reconstituted with alpha-4 methyl mutant is substantially attenuated relative to control level, this purple line. So these data indicate SMID5 via alpha-4K22 trimethylation promotes xenograft tumor growth. Okay, let me sip of cup of water. Next, to better understand the role of SMID5 in vivo, our collaborator generate 2 different robust mouse model for gastric cancer, and one based on CDH1 mutation, the hereditary mutation, the common mutation found in the gastric cancer. And second, based on the SNP complex commonly found in sporadic gastric cancer, here represented by ARID1A. And they were combined with canonical pathway like KRAS or P53 or P10 mutation. And then they designed to be controlled under gastric specific on-action 10 promoter, and then regulated by Cre recombinase with tamoxifen. And they crossed with Smith-5 knockout mice. And they are now referred to KPPC or KPPA model. And then we studied the survival rate. As you see here, survival curve of both mouse models. Smith-5 depletion increased the lifespan over 70% of KPPC model, 45% in KPPA model. And more interestingly, control mice developed metastasis, more than 50% of cohort with lung, liver or lung metastasis, and with peritoneal carcinomatosis. But strikingly, Smith-5 knockout mice, they developed zero metastasis. As you can see here, this image, compared to the control, the tumors in Smith-5 knockout, they look like confined and within the stomach tissue. And consistently, I'm showing here a representative H staining image of the KPPC model, and indicating their notable cancer cell invasion here in the control. They get through the basement membrane. But those in Smith-5 knockout mice, the tumor look like confined and benign tumor. So we concluded that Smith-5 may function in driving gastric cancer malignant progression. And we further test that idea, and we developed a metastatic PC model. There is a form of metastatic gastric cancer referred to peritoneal carcinomatosis, in short, PC. Once it penetrates the peritoneum, the lining, the organ in like stomach or other organs, and it makes very difficult prognosis and account for more than half of gastric cancer patients. So we generate PC xenograft model where NOXI4 metastatic gastric cancer cell lines. We directly implant the NOXI4 cancer cell lines into the peritoneum, and we monitored bioluminescence of the tumor growth, the cells expressing the luciferase reporter. And we hypothesize maybe inhibiting Smith-5 pathway may increase the efficacy of the known anti-cancer drug. So we did the drug screening with the known drugs, like in combination, to see if any drug have the synergy with Smith-5 depletion. And then the top hit was the PI3K mTOR pathway inhibitor called omeprazole. And we did the combo therapy, like a dual therapy here. And here I'm showing the survival curve. The Smith-5 depletion increased like a 40% of lifespan, and omeprazole treatment 65%, And then combining those two treatments, there was a synergy of increase the lifespan. And then last, we further leverage this combo therapy combined with immunotherapy called CAR-T. And we use the mesoderine, a common antigen in gastric cancer, to create the mesoderine reactive CAR-T cells, and use the same PC model. We injected the CAR-T into the peritoneum, combined with omeprazole treatment and SMID5 depletion. And then we named triple therapy. And looking at the survival curve data compared to CAR-T alone, CAR-T omeprazole dual treatment increased modest increase in the lifespan. But surprisingly, if you add SMID5 depletion, like a triple therapy, entire cohort of mice or alive with cancer-free survival, it completely cured this metastatic cancer. And here, I'm showing the tumor growth monitoring data. You see at day one, we injected the tumor in the peritoneum. And in control mice, all the mice died before day 35. They died. But in triple therapy mice, you see all tumors that disappeared, and they all survived even at the time of end of experimental points. So this is a pretty exciting finding. This is a pretty harsh metastatic cancer model. But it's great to see this triple therapy combined with SMID5 inhibition cure the disease. So it is early preclinical result, but we need, I think this is a great potential to develop the SMID5 inhibitor. Let me summarize my research on SMID5. SMID5 is the enzyme that methylates alpha-40, and they increase in cancer patients. And SMID5 regulates mRNA translation of oncogenic programs and impacts gastric cancer progression. Lastly, we show that SMID5 inhibition combined with other known therapies could be a new generation treatment for the gastric cancer. This is my final slide I prepared today. Thank you.